is our medical category finalist, NETrolyze, a novel immunotherapy for triple-negative breast cancer. And we have with us Sam Carrier and Priyan Virapalli, who will be presenting the idea of the medical category.
Hi, I'm Priyan. This is Sam, and we're here from Extrinsic Immunity Therapeutics to talk about NETrolyze. Before we begin, to sort of level set, I want to reframe what cancer is. We typically think of cancer as a process of uncontrolled cell division, and that sort of inevitably brings up this idea, this image of size. And sort of intuitively, viscerally, we react to larger tumors as being worse, smaller tumors as potentially being better. But if you actually look at what accounts for mortality in cancer, it's actually not just the presence of a tumor, it's tumor spread. It's the process of metastasis. And metastasis remains the underlying cause of death in about 90% of deaths from solid tumors. And that statistic is sort of captured here. So if you look at the 5-year survival rate for someone who's diagnosed with localized breast cancer, that tends to be around 90%, whereas people who are diagnosed with metastatic disease, it's quite a bit lower. But if you look at the therapeutic landscape, most, if not all, of the therapeutics that are on the market target the actual process of cell division. So the goal is to kill dividing cells. And the act of trying to target metastasis is still not a therapeutic possibility. And that's really where NETrolyze comes in. So a lot of our work is based on experiments that were done within the last 7 years. And what basically has been found is that in a mouse model of triple-negative breast cancer, if you implant these tumors within the mammary fat pads or the breast tissue of the mouse, these triple-negative breast tumors will sort of attract neutrophils, one of the early responders in the immune system. And these neutrophils release a material within the tumor microenvironment that's referred to as NETs, neutrophil extracellular traps. And this material, for whatever reason, seems to promote metastasis. What's interesting is that this initial work found that if you prevent neutrophil infiltration, if you prevent NET formation, or if you just degrade these NETs locally, all of these approaches are successful and sufficient to prevent or delay metastasis. And that's really where NETrolyze comes in. To sort of emphasize what the value proposition here is, if you think of patients who are diagnosed with a tumor, the average time between tumor detection and surgery is about 40 days. It can feel like quite a bit longer if you're the one who's been diagnosed. I had my mother go through this, so it feels like quite a bit of time, even if it's just 10 days between diagnosis and the beginning of intervention. There remains a lack of existing therapies, targeted therapies that can target tumors, especially in triple-negative breast cancer. And so most patients actually end up cycling between multiple therapies. So you'll be on one therapy for some amount of time, you'll develop resistance, and then you'll cycle to a new therapy. And the median duration of the first-line therapy is between 4.4 and 6 months. Second-line therapy tends to be a little bit less. And so what we have basically done is we've formulated an FDA-approved small molecule drug that's currently used for other indications into a slow-release gel. And that gel we can then inject near the tumor site, and it leaches out this drug. And this drug then degrades NETs locally. And the sort of broad objective is to target the tumor, extend the treatment window for each patient, and ultimately to save patient lives.
So what gets us really excited about NETs is actually the impact it can make, so the broad impact and really the number of people it can reach. So up to 70% of women with triple-negative breast cancer could directly benefit from NETrolyze. And we know that this is the right product for the right market, really based off of 3 pillars. So the first is this blockbuster revenue potential. NETrolyze contends to be north of a $1 billion market opportunity in a growing market. It's high reimbursement potential, so because this is a first-in-class drug, insurers are more likely to pay for it, which means there's high value in access. And lastly, accelerated approval. So because triple negative breast cancer is seen as a high unmet need area by the FDA, almost all therapeutics that have walked this path before have been granted some accelerated approval. Talking about commercialization, there's some highlights that we want to point out. So first, we see this as having a very broad customer base. Really, any general hospital that does cancer screening services would be a great proponent for NETrolyze. We're going into an untapped industry. We really are a one-of-one NETs oncology product. We also integrate seamlessly with the standard of care, so we work hand in hand with existing therapeutics to really not require a huge overhaul of the system. So upon commercialization, immediate impact and protection. We have IP around the gel, as well as some other IP around diagnostics, and we also have licenses to exclusively commercialize this IP. When we look at the competitive landscape, so we want to show how we're differentiated between what's out there and what is currently the standard of care. And so there's really two subsectors of competitors we want to look at. The first are these direct competing compounds. And so these are Pulmozyme or Jubilant's PAD4 inhibitor. So these are still in the clinic or being tried for new indications. But we really kind of beat out these competitors for three reasons. The first is that they are non-tumor targeting. So any effect they have on NETs is indirect. The third is that they're for a different therapeutic area, so not cancer. They're looking at cystic fibrosis, investigating autoimmune disease. And third and finally, which is the biggest one, they're not designed like we are to leave the immune system unimpaired. And so NETrolyze beats out the competition in those three ways. And then we want to look at the existing landscape right now, the standard of care. So these are therapies that are on the market, so Keytruda, Talzena, and what we really find is that these aren't really competitors. They're more synergistic compounds, things that can be co-formulated with NETrolyze to actually make our therapeutic and their therapeutic work better together. And we're currently doing research right now to prove out that hypothesis. The leadership team who's actually going to bring this product to the market, so Priyan and I are co-founders. Priyan brings 10 years of research and development in the NET space. He's an American Cancer Society Fellow and a PhD in biomedical engineering. Myself, background's in biochemistry. I was in industry for drug development for a while. Now I'm getting my MBA at Michigan. We also have a great leadership team of leading biostatisticians, senior scientists, clinicians at the University of Michigan, as well as a full advisory board who's brought multiple drugs from academia into industry. And we also have a CMC partner, so we are negotiating with Latitude Pharmaceuticals who can actually make this, scale this drug, and actually realize its potential. Short-term roadmap as well, so we've built out key milestones for the next 2 years to get to an IND phase that is submitting paperwork so it can be first in human. So right now, we are working on our first pilot. It's in the lab right now. We have mice with tumors in them right now being studied and analyzed. So these are our efficacy studies. On the horizon as well, we have pharmacology and toxicology studies. But looking more long-term, how we're going to create a better future is a 7-year roadmap. So like I was saying today, we're in preclinical. We're really working with non-dilutive sources, prize money from things like Create the Future, non-dilutive grants like NCI and NSF to build out and accelerate our R&D with the ultimate goal within 7 years to get to phase 2, which is kind of the inflection point where we hope to partner with an established player, a large pharmaceutical company, to really expand and realize its potential. So with that, thank you again for having us in New York. We'll open it up for questions. Thanks.
Questions? Yes. Start.
Always questions. So the mechanism of action, you explained that briefly. That's fascinating. I think it's a very new field. Maybe not for you with the NETs, but it's obviously a physiological evolutionary mechanism, right?
Right.
And so I think it has antimicrobial effects and some effects. So the question would be if you disrupt it or if you stop it or influence it, can you speculate about side effects?
Yeah, so this material, it turns out, I don't know if I did. So the material, it turns out to be beneficial in wound healing, and it tends to be beneficial to prevent or delay infection, which is why our goal is really to target that material only around the tumor and not to suppress sort of systemic immunity. Within the tumor, we have hypotheses based on sort of evolutionary biology for why it might show up in these tumors. The tumors, so it doesn't just apply to breast tumors. We've seen it with other tumor cell lines as well, and we're sort of exploring that space now. The tumors that seem to react to it are from tissues that are outward facing, so facing the inner surface of the lung, the outer surface of the ductal surface of the salivary gland. So we think maybe these tissues are sort of evolved to recruit immune cells when needed, and this is maybe a mechanism that the tumor hijacks to then allow these tumor cells to kind of escape through either new vascularization or other mechanisms that are still kind of unknown.
Next question.
Just a couple of things. Your competitive landscape slide was very interesting. Okay. It looked like you were identifying other potential cancers that would be able to be treated with this molecule. Can you comment about that? And whether or not you would need a different type of molecule, or you could use the exact same molecule on the different ones. And then following on, I'll get this one in. The FDA, have you talked to them yet? And if so, are they targeting you for fast tracking in any way?
We actually just sent emails to the FDA to start scheduling these kind of early meetings. So we haven't heard back yet, but this was about a couple of weeks old. The first question, so in terms of the exact molecule, the molecule works through a charge interaction, so it's not based on enzymatic activity, it's not sensitive to a lot of things that those sorts of molecules might be. And so, for example, the mechanical properties of the pancreas will be different from the mechanical properties of breast tissue versus other tissues, right? And so we may need to work on the gel formulation to kind of tweak it for the type of tissue you're injecting into. But we think the molecule itself should be okay.
And do you think the organs are going to be tolerant of the gel injections?
So far they have, at least in the breast experiments we've started in mouse models, we haven't seen any adverse reaction to the gel.
And have you looked at anything else other than breast then? Or
in vitro we've looked at pancreas, salivary gland, ovarian, lung, sarcomas, but in actual animal models we focus just on the breast tissue for now.
And the results in those other tissues, did the tissue samples?
So what we're looking for, what seems to happen in the tumors that sort of metastasize when this material is present, the tumors tend to recruit more and more neutrophils. And what we find is in vitro, when you culture tumor cells with this material, they upregulate the production of neutrophil chemothractins, inflammatory cytokines like GCSF, and we think there's kind of a positive feedback loop that might be driving this influx. So in vitro, to identify other tissues that might respond, we're basically culturing those tumor cells with the material to see if they upregulate the production. And there's subsets of all of these cancers that react in the same way.
I think we're done with the questions. Thank you.
Thank you.